Welcome to today's week 7 tutorial class of experimental biotechnology. Hope my screen is clearly visible and I am clearly audible. So I am Ananya Chatterjee, PhD student of IIC Bangalore, and I will be your teaching assistant for this course. Instructor of this course is Professor Vishal Trivedi, Professor of IIT Guwahati. So till week 7 tutorial class has been completed and we have so far discussed assignment 1 to 5. So and also assignment 6 part 1 and part 2. Today we'll be discussing assignment 6 part 3 and whichever deadline is over, and also for week 7 possible questions from previous year we already discussed in last class. Today we'll be discussing some possible questions from previous year's assignments of week 8, that is cell biology. So here this is the YouTube link of all tutorial classes we conduct, and this is the folder link of tutorial slides. So we'll be starting with assignment 6 part 3, and before that, if you have any particular question, you can ask in the comment section. You can convey your query. So we'll be starting with first question of assignment 6 part 3, which is: which of the following chromatography techniques exploits the shape complementarity? Options: affinity chromatography, ion exchange, gel filtration, hydrophobic interaction chromatography. So the answer will be affinity chromatography. So affinity chromatography relies on the specific interaction of a solute with a ligand. Okay, so the ligand is attached to a support resin, so like this. So in affinity chromatography, the ligands will be attached to a support resin. The specific interaction between the solute and the ligand is strong, similar to substrate enzyme interaction. So this affinity may be based on molecular size and shape also. So shape complementarity comes from affinity chromatography. So in this case, the force controlling the interaction can include ionic, covalent and or hydrogen bonds. Most commonly, an antibody is used as a ligand which recognizes the target protein with lock and key precision. Affinity chromatography is the most expensive chromatographic method, since often a highly purified protein, the antibody, must also be manufactured before the target protein. Owing to the highly specific binding affinities, affinity chromatography can be used to give highly purified proteins in a single step. So the shape complementarity comes from the affinity chromatography. Question 2: given a receptor and two ligands, ligand 1 and ligand 2, where a receptor is forming a complex between R and L1, and which complex is very tight, than RL2. Which of the following statement is true for Kd? What is Kd? Kd is dissociation constant. Okay. So one complex is very much tight and another complex is very much loose. That means one complex dissociates slower and another complex dissociates faster. So the answer will be the complex which is dissociates slower, that means which is very tight, that Kd will be low. That means Kd of RL1 will be lower than Kd of RL2, right? Answer will be B. Affinity chromatography is often preferred for separating molecules with similar molecular weights, opposite charges, strong hydrophobic interactions, specific binding affinities. Yeah. So in affinity chromatography, we are relying on binding affinities, right? So the so it's often preferred for separating molecules with specific binding affinities. Yeah, so the KD, which is the definition of KD, when receptor and ligand complex formation is happening, a mutual interaction between a ligand and receptor forms ligand receptor complex with a dissociation constant of KD, where KD will be RL divided by R, complex of RL. Okay. Next question. Using GST glutathione interaction is an example of bioaffinity chromatography, pseudo affinity, diaffinity, metal affinity. So GST glutathione, because there is a affinity between GST and glutathione, so this comes under bioaffinity chromatography. So in this type of affinity chromatography, where biomolecules are used as receptor on matrix, and it exploit the biological affinity phenomenon such as antibody antigen, that's why the name comes as bio. In addition, enzyme substrate or enzyme inhibitor is also belong to this class. So here example is GST glutathione. So lysine coupled matrix can be used to purify which of the following molecules? Answer will be rRNA. So we provided one list. These are the different receptors, and this is matrix containing receptors for ligand present on protein. So for lysine, affinity towards protein ligand rRNA. Okay. Next question. Carbodiimides reacts with matrix containing what group to form what group? So we have read that it forms, so it reacts with a matrix containing carboxyl group and to form isourea ester. So it in the matrix, it has polysaccharide matrix, so it's binding with carboxyl group, and after binding with the ligand, it is forming isourea ester. Okay, so it's binding with carbamidite, then what is forming? COO, CNH, NHR. So it is isourea ester. So there is a urea group, and so here it's called isourea ester because there is a ester linkage also. So that's why carbamides reacts with the matrix containing carboxyl group to form isourea ester. So the activated matrix is then allowed to react with the receptor molecule containing carboxyl or free amino group. Receptors are coupled to the matrix by a secondary amine linkage. Answer will be C. Affinity chromatography column is equilibrated with buffer containing 0.5 molar NaOH. NaCl. Equilibrated with buffer containing 0.5 molar NaCl. That is, yeah. How does chaotropic salt helps in elution in affinity chromatography? Options: it changes pH of the mobile phase, it partially denatures the receptor, it denatures the ligands, it removes both ligands and receptors from the matrix. So how does chaotropic salt helps in elution in affinity chromatography? So what is happening? There is ligand and receptor. Okay, there is receptor and then ligand. Okay, so what they do, they basically partially denatures the receptor so that receptors and ligand interaction gets disrupted and elution happens. What is used to wash the affinity column after use? So after use, you can wash the affinity column using guanidine hydrochloride to denature all the proteins associated with the column so that the column gets cleaned up. Okay, you can use guanidine hydrochloride. Also ACl you can't use, but 1 molar NaOH you can use, and also 20% ethanol you can store, but for washing you have to use this guanidine hydrochloride. So the answer will be that. What What are used for coupling with antigen for bioaffinity chromatography? Cephalus beads. Okay. CNBR activated matrix reacts with free amine groups on proteins or peptide ligands. In affinity chromatography, which fusion protein tag is commonly used for protein purification and can be specifically eluted using reduced glutathione? So, we have read about all these things in week 5 tutorial classes in details. So, if you are thinking about the concept, we have already talked in the class 5th tutorial session. So, answer will be GST tag. So, in this case, we use reduced glutathione to elute. What is used to remove GST tag from purified fusion? If there is a thrombin protease site, you can use thrombin. Nickel anti-affinity chromatography is most suitable for the purification of protein with histidine tag. Okay. Which of the following elution conditions is commonly used to release histidine tagged proteins? So we have talked about this because imidazole is a similar potential like histidine tagged protein. So imidazole will bind with the, like, it has higher affinity, so that's why we use imidazole to elute those histidine tags protein. Okay. So week 6 assignment we have discussed part 3. We need to discuss week 7 assignment also, but that is still deadline is there. So if you have any specific questions, you can let me know, or we'll discuss week 8 possible questions which you might have in your next class, next assignment. This is from previous year's questions. So we will solve these assignments after discussing at the end of today's class. Before that, we'll discuss the cell biology course to understand this concept clearly. So yesterday, like the previous day, we were discussing cytokine array. So we left little bit. We'll discuss this and we'll start with cell biology. So cytokine array, what is that? So carefully selected capture antibodies have been spotted in duplicate on nitrocellulose membrane. The sample and antibody mixture is then incubated with mouse cytokine array membrane. So what you do in the cytokine array is like very much like ELISA. So this is a sandwich ELISA. You can see, so here antibody is there, and another like antibody with a sample is there, and another antibody is capturing it, and then the secondary antibody is detecting those antibody. So here you can see this cytokine array chip. So this is basically cytokines array chip where cytokines are attached. So now, so for the materials you need to have mouse cytokine array, then some buffer, then detection antibody cocktail, streptavidin HRP, some reagents, and multi-dish and transparency overlay. So the possible is sample collection and storage. Remove particulates by centrifuge. So at first the cell culture supernatant you take, and then remove particulates by centrifugation. Assay immediately or aliquot and store. So you can do the cytokine array assay immediately, or you can store the sample at -20 for future use. So you should avoid repeated freeze-thaw cycles because those proteins might get degraded. Now cell lysates, what do? You have to solubilize cells and then in a lysis buffer, pipette up and down to resuspend, then rock the lysates gently at 2 to 4 degrees Celsius for 30 minutes, microcentrifuge at 15,000 G for 5 minutes, and transfer to the supernatant into a clean test tube. Quantification of sample protein concentration using a total protein assay is recommended. Serum, allow blood sample to clot for 30 minutes at room temperature before centrifuging for 15 minutes at approximately 2000 G. Remove serum and assay immediately or aliquot, and store samples at -20 degree. Plasma, collect plasma using EDTA or heparin as an anticoagulant. Centrifuge for 15 minutes at approximately 2000 G within 30 minutes of collection. Assay immediately or aliquot at -20. Tissue lysate, excise tissue and homogenize in PBS with protease inhibitors. After homogenization, add Triton X100 to a final concentration to 1%. Freeze samples, then thaw and centrifuge to remove cellular debris. Reagent preparation: mouse cytokine array, 4 nitrocellulose membranes each containing 40 different anti-cytokine antibodies printed in duplicate. Handle membranes only with gloved hands and flat tip tweezers. Detection antibody cocktail: 1 vial of lyophilized biotinylated antibodies. Before use, reconstitute the detection antibody cocktail with 100 microliter of deionized or distilled water. What is 1x wash buffer? Here you have to make add around 40 ml of 25x wash buffer concentrated to 960 ml of deionized water to prepare 1000 ml. So you have to, whatever your buffer concentration was, you have to make 1x buffer, and then chemi reagent mix. Chemi reagent means 1 and 2 should be mixed in equal volumes within 50 minutes of use. Protect from light. 1 ml of the resultant mixture is required per membrane. Discard any remaining after use. Okay. So the assay procedure is: bring all reagents to room temperature before use. Keep samples on ice to avoid contamination. Wear gloves. Prepare all reagents and samples as directed in the previous sections. So now you have to take array buffer into each well of the 4 well multi-dish to be used. Array buffer 6 serves as a block buffer. Using a flat tip tweezers, remove each membrane to be used from between the protective sheets and then place in a well of 4 well multi-dish. The number of the membrane should be facing upward. Incubate for 1 hour on a rocking platform shaker. Orient the tray so that each membrane rocks end to end its well. While the membranes are blocking, prepare samples by adding up to 1 ml of each sample to 0.5 ml of array buffer 4 in separate tubes. Adjust to a final volume of 1.5 ml with array buffer as necessary. Add 15 microliter of reconstituted mouse cytokine array antibody cocktail to each prepared sample. So this is the same assay procedure we are discussing. So now you have to take aspirate array buffer from the wells and add sample or antibody mixture. Now again you have to incubate on a overnight on a rocking platform. Carefully remove each membrane and place into individual plastic containers with 20 ml of wash. Now you have to do the wash. Wash each membrane for 10 minutes in a rocking platform. Then you have to dilute the HRP in an array buffer using dilution factor of the vial level. Carefully remove each membrane from its wash container. Allow excess buffer to drain from the membrane. Return the membrane into the 4 well multi-dish containing the diluted streptovirine HIP. Cover the wells with the lid. Incubate for 30 minutes at room temperature on a rocking platform shaker. Carefully remove each membrane from its wash container. Allow excess wash buffer to drain from the membrane by blotting the lower edge onto paper towel. Place each membrane on the bottom sheet of the plastic sheet protector. Carefully, so now the thing is that carefully you have to cover the top sheet of the plastic sheet protector. Gently smooth out any air bubble. So you have to add these things, and so you have to make sure that any air bubble is not there, and then you have to image then you have to position the paper towels on top and slides of plastic sheet protector containing the membranes and carefully squeeze out excess reagent. Then the top plastic sheets protector you have to remove, and you have to carefully lay on absorbent lab wipe on the top of membrane to blot off any excess reagent. So this is a this is from the reference. So this is reference for this is untreated and treated. As you have added, wherever you have added antibody against your protein, so those things are light up. So yeah, those things are getting lighter. So now the data analysis part is that the positive signal seen on developed film can be quickly identified by placing the transparency overlay template on the array image and aligning with the pairs of reference pod in 3 corners of each array. Reference pods are included to align the transparency overlay template and to demonstrate that the array has been incubated with streptovirine HRP during the assay procedure. Pixel densities on developed X-ray film can be collected and analyzed using a transmission mode scanner and image analysis software. You have to create a template to analyze pixel densities. Export signal values to a spreadsheet file for manipulation in a program such as Microsoft XL. Determine the average signal of the pair of duplicate spots representing each cytokine. Subtract an average background signal from each spot. Compare corresponding signals on different arrays to determine the relative change in cytokine levels between samples. So these procedures are very much similar to Western blot ELISA. So detection of interaction of antigen antibody detection of interaction of antigen and antibody. So we'll be starting today's class by discussing cell biology. So now cell biology part 1 has been live. Now cell biology part 1 has been live. So here there are different kinds of cells. You can see prokaryotic cells, eukaryotic cells. So this is the animal cells, this is the plant cells, and there are instant fungi also. So growth and multiplication of host organism used as expression system requires a suitable biochemical and biophysical conditions. The biochemical conditions can be provided by the use of various nutrient medium. Depending upon the special needs, different types of media have been developed for expression system to achieve growth, multiplication and overexpression of protein. So like you need for the growth medium, what do you need to grow a cell? Proteins, carbohydrates, lipids, DNA, RNA, amino acids, sugar, fatty acids, nucleotide, building block, energy, genome, different like atoms, CHO, phosphate, etc., minerals, vitamins. Microbiology medium constituents. So those are constituents and those are the source. So amino or nitrogen, source peptone, protein hydrolysate, infusions, extracts. Growth factors, blood, serum, yeast extract or vitamins. Energy resources, sugar, alcohols, carbohydrates. Buffer salts, phosphates, acetates. Mineral salt you can get from phosphate, sulfate, magnesium, calcium, iron. Selective agents you can get chemicals, antimicrobials and dyes. Indicator dyes you can get from phenol red, neutral red. Gelling agent, agar, gelatin, etc. So this is some composition of selected media for bacterial growth. So bacterial growth, M9 minimal media. What is the composition? 0.6% disodium hydrogen phosphate, 0.3% potassium hydrogen phosphate, 0.05% sodium chloride and 0.1% ammonium chloride. Application for cultivation and maintenance of E. coli strains. Another media, M63 minimal media. So the minimal media. So these are the compositions you have to know. It's not like you have to remember, but you have to have an idea that what are there. So here ammonium sulfate are there, potassium dihydrogen phosphate are there. So you can see that from where your minerals, nitrogen, energy sources are coming. So the application of M63 minimal media is for cultivation and maintenance of E. coli strains. LB what are there? Peptone, yeast extract, NaCl for E. coli growth. LB Lennox broth, it has 1% peptone, I think Hari Narayana has question, what are purines? Purines or proteins? What do you... Purines is nowhere or protein? What do you want to write? Purines. You are asking question from the slide I am teaching, or 8. Okay, we'll discuss week 8 after some time. Week 8 means cell biology. Yeah, week 8 means the thing we are reading now. We'll come to that point. Because purines is, so this is specifically a protein which forms the pores, and also yeah, so we'll discuss that. I'm not able to understand that from exactly where, which context you are telling, so I will get back to you with that, because we have seen different channels now, like hydrogen channels, like water channels, so aquaporine. So these are different proteins which forms the pore in the cell. So I think that's why you are asking. That's the thing. We'll get back to that. So look, whatever media we are now discussing, they have asked like direct questions from that. So you have to understand that for which cultivation you use those. You have to remember a little bit. So the specific questions, can you tell? The question is where is purines present? You can tell that, is there any options of cell wall or cell membrane? Then it will be cell membrane. Okay, so these are the different growth media. SOB media, peptide is present, yeast extract, NaCl and KCl, magnesium chloride, to make high efficiency competent cells. SOC media, SOB plus 20 millimolar glucose, growth of competent cells. YT broth, yeast extract plus tryptone. Uh, Terrific broth media, peptide, yeast extract, those are present for protein expression and plasmid production. Then TYGPN media, tryptone, yeast extract, glycerol, these are present. So microbiology medium preparation. For preparation of bacterial media, dissolve the components in 1 liter of distilled water. Cover top of the flask with cotton plug and autoclave the solution at 120 degree for 20 minutes. So all the media you have to prepare, you have to dissolve that in your required amount of water. Okay, it's not like the 1 liter. If you need 20 ml, you need to dissolve in 20 ml. Okay, and then cover the top with a it's very well covered with cotton plug, and so that the aeration also is there, and autoclave the solution at 120 degree at 20 minutes. Precautions while making microbiology medium: ratio of medium volume and cultural flux. So aeration is needed, so for that you have to make that, like in 500 ml flask you can't make 400 ml media, right? You can make around 150 to 200 ml media, like that. Medium components are hygroscopic, and while waiting, avoid moisture stored in a cool and dry place. While autoclaving, open the autoclave only after it's cold. Avoid charring of media components. Solid media should be poured in plates once it's cooled to 50 degrees Celsius. The various antibiotics or nutrient supplements should be added to the medium when the temperature is less than 50 degrees. Okay. So these are different yeast media. Selected growth media for yeast expression system. This is CSM media. So composition is CSM without tryptophan, yeast, nitrogen base, ammonium sulfate, glucose agar. Application for making agar plates that enable the growth of Saccharomyces cerevisiae. YPD broth, yeast extract, bactopeptone, dextrose. Commonly used yeast media for maintenance and propagation of Pasteuris and cerevisiae. YP gal, bacto yeast extract, bactopeptone, galactose, bacto agar. Standard medium of cerevisiae omitting glucose repression. Standard minimal media or yeast nitrogen base. So yeast nitrogen base with ammonium sulfate and without amino amino acids, and dextrose plus any amino acids or nucleotides. So these are the different constituents. Method of preparation: as per as the media composition, constituents are in 950 ml of water on autoclave. Allow media to cool 50 degree and then add 550 ml of filtered sterile 40% dextrose so that the final concentration become 2%. Adjust the final volume to 1 liter if necessary. Now we'll come to mammalian cell culture media. So recipe of mammalian cell culture complete media. These are the components. This is composition. So DMEM composition around this 13.4 gram per liter. This amount you don't have to remember, but you have to remember that it has DMEM, sodium bicarbonate, fetal bovine serum, then antibiotic unit. So preparation of cell culture media. To explain the method of media preparation, we are taking the example of DMEM media. Add 13.4 gram dry powder medium to the water. We are taking the example of DMEM media. Add 13.4 gram of dry powder medium to the water and mix and dissolve it completely. Add sodium bicarbonate, mix completely and adjust the pH to 6.9 to 7.1 using 1 normal LNH or 1 normal ACL. Finally, add cell culture grade water to the media to bring it to the final volume. Sterilize the solution using sterilized membrane filter with a pore size of 0.22 micron. Supplements such as antibiotic serum can be added to the sterilized solution using aseptic technique. Okay, so this media can be commercially bought also. You can buy the whole media commercially, also you can make. So generally people use it commercially bought available media. So you have to have this pH set. pH should be adjusted. Filtration should be performed at low speed. Serum heat inactivated. Antibiotics contamination checking. Selected growth media for insect cell culture. So grasses insect media supplemented growth media. Composition unsupplemented media actalbumin hydrolyzed histolate. Application growth of Spodoptera SF29 cells or SF9 cells. Hinks TNF FH insect media. So this is supplemented with grasses, again L-glutamine. So for the culture of cabbage looper, IPL 41 insect media, IPL 41 media, calcium chloride, L-glutamine, TC100 media, L-glutamine, sodium bicarbonate, and so on. Now we'll discuss the second part of the cell biology. Second part means the week 8 second part. So this is fractionation of prokaryotic cells. So what are the components basically? So here you can see. So look, you were talking about porins, no? Look, in the membrane, as I was telling you, the porins are present in the outer membrane. Okay. Hari Narayana. Yeah. So in the prokaryotic cells, porins are there. So this is basically present in the outer membrane. So where lipopolysaccharides are also there. So you were telling that it's not available in the lecture notes, but it's available. See, okay. So and also it consists of lipoprotein. This is a periplasmic space. This is peptidoglycan. This is so the whole thing is peptidoglycan. It has okay, so it has in the membrane, it has inner membrane and outer membrane, and so this is the cytoplasm, this is the gram negative cell wall. So where you can see subcellular localization, periplasmic space, total number of proteins 129. You don't have to remember these numbers. Outer membrane also has some amount of proteins, extracellular, inner membrane, cytoplasm. Okay. So isolation of periplasmic fraction. So harvest the bacterial cells by centrifugation of 3000 G for 20 minutes. Discard the supernatant and carefully remove the few drops of liquid with a pipette. Gently resuspend the pellet in a 1 ml of TSC buffer with a wire loop. 30-minute incubation on ice. Transfer the cells in microcentrifuge tube and centrifuge at maximum speed. Transfer the supernatant to a new centrifuge tube. This supernatant constitutes the envelope extract and periplasmic fraction. Fractionation of eukaryotic cells, if you fractionate you will get cell, chloroplast, cytosol, mitochondria, plasma membrane. Different types of centrifugation are there at different speeds. Okay, this is a Beckman ultra centrifuge, this is tabletop centrifuge. So there cubota also you will find. So centrifugation, you can do differential centrifugation, you can do density gradient centrifugation. So differential centrifugation, what is that? Based on the differences in sedimentation rate of the biological properties of different size, shape and density. So differential centrifugation is based on different size, shape and density. Okay, so this is a time of centrifugation. You like as 1 hour, 2 hour or like that. As soon as you are increasing the time of centrifugation, the sedimentation rate is also you know it's increasing. Okay, and this is a centrifugal fill. So you can see the smaller one, then bigger one, and another more bigger one are getting sedimented at different layer and different speed. So this is large size, this is medium size, this is small size. Now separation by sedimentation, you can, so you have some materials, iron, stone, again cotton, like that, and their weights are different, 100 kg, 30, 10 kg, 8 kg, 1 kg, and like that. So what it will do, it will, the separation will be based on the weight. Okay, so whatever, like it's okay, separation by sedimentation. Okay, so this separation will be based on the density, and also like you can see that sedimentation higher density. Okay, so whatever density is higher, like irons will be sedimented at the very bottom. So like if you are doing a sedimentation based on density, so it will be in that layer where the density is matching with its density. Okay, so what we are seeing here, there is irons, here so we can see stones of different weights but the density is similar, and here it's cotton. So this is the principle of sedimentation. Okay, so here again you can isolate proteins, DNAs, like that, based on the density. Now example of isolation of cell organelles. So you have isolated your tissue and you have to mix with buffer and you have to homogenize it. Then you will do the whole cell connective tissue, so you will then ultracentrifuge it. So after, what will happen, at first at this much G the nuclear cytokeratin will get precipitated, then you have to increase the G speed, then mitochondria, lysosome, peroxisome you will get. After that, after increasing furthermore, plasma membrane, ER fragments, small vesicles you will get, and then after that, after increasing more G force, you will get ribosome, viruses and macromolecules sedimented at the down. Okay, so cell fractionations you can do by centrifugation. So this is muscle cell, you have to homogenize, you will get tissue homogenate. After tissue homogenate, you will get nuclei and cell debris and supernatant. So supernatant, you have to do gradual centrifugation at different gradual increasing speed, and at specific speed you have to isolate your organelle of interest. So repeated centrifugation at progressively higher speeds will fractionate homogenate of cells into their components. In general, the smaller the subcellular component, the greater is the centrifugal force required to sediment it. Densities of biological materials. So microbial cells density is around 1.1 to 1.15, mammalian cell density 1.04 to 1.10, organelle density 1.10 to 1.60, protein density 1.30, DNA 1.70, RNA 2. Density gradient centrifugation. So this is zonal centrifugation, sucrose density gradient. So you have to make a density gradient, like from top to bottom or from bottom to top, the density will be decrease. From bottom to top the density will be decreased like that. So what will happen, you have made a sucrose density gradient and you have added the your sample on top of the this density gradient. So this is after certain time, 0 hour, 30 minutes, 60 minutes, 120 minutes and so on. So you will get to see that your samples are getting sedimented and those are getting like differentiated. Particles migrate according to S value. This is S value is a wet bulb unit. So particles migrate according to that, and then it will migrate to its own density zone, and then you will collect those fractions like that. So you will get you are getting a fraction as you will collect those your desired fractions from that. So what you can do, you can collect those desired fractions from syringe also and from pipettes also. Manual collection you can do from pipette, or automatic fraction collectors are also there, and freezing and slicing, so you can freeze the whole sample and then slice, cut through it. These are the different possible process for this collection. So this is all for week 8. Now we will discuss possible questions of week 8 cell biology from previous year. So the first question is, what happens to the color of mammalian cell culture DMEM media when pH of the media decreases? pH of the media decreases means if the pH is the cell culture media pH should be around 7.4. So if pH is decreases from 7.4 to मान लो 4, what will happen? There will be acidity, so the color of phenyl red will be towards yellow, so changes to yellow. The medium component responsible for osmotic balance is osmotic balance, so it will be NaCl, right? During bacterial media preparation, antibiotics should be ideally added to cell culture media when just before autoclaving the media, when temperature is just above 90 degree, when temperature is less than 50 degree. The answer will be when temperature is less than 50 degrees Celsius. The pH of the DMA media is around 6.9 to 7.1. It's around 7 or 7.4 like that, like physiological pH is around 6.9 to 7.1. The medium used in cell fractionation should not be hypotonic. It should not be hypotonic. It can be cold. It should be buffered. It should be isotonic, but it cannot be hypotonic. There will be osmotic shock if it's hypotonic or hypertonic, right? So the treatment to remove the cell wall from plant is, you can use cellulase. Then it will degrade the cellulose from the cell wall. The centrifugal force and time needed to carry out separation of the mitochondria from a whole cell lysis, you have to remember that it's around 15,000 G, 15 minutes. While preparing a mammalian cell culture media, a student makes mistake and ends with getting contamination in the media as given below. Identify the mistake for the observed contamination. Use of 0.22 micron filter instead of 0.45 micron filter. Use of 0.45 micron filter instead of 22. Not autoclaving the media. Use of biosafety cabinet for filtration. So identify the mistake of the observed contamination. So while making a media, they made a mistake in getting contamination in the media. So if they would have used 0.45 micron filter, then those larger particulate would have passed into the membrane, into the media. So this is a mistake that he or she has used 0.45 micron filter instead of 0.22 micron filter. The M63 and M9 media are used respectively for the cultivation of E. coli strains. In the isolation of periplasmic fraction of the prokaryotic cell, which substance or action is used to give osmotic shock? For osmotic shock, you have to have TAC buffer followed by centrifugation at 6000 G. In a certain study performed by a group of students on an unknown cell type, yielded the following fraction. A, B, C and D. Sorry. A, B, C and D, and 4 steps of centrifugation at 600 G, 10,000 G, 1 lakh G and 3 lakh 20,000 G. The different organelles present in A, B, C and D fraction is, so if you remember, at very low speed 600 G your nucleus will get separated out, around 10 to 15,000 G your mitochondria will be coming, then 1 lakh G ribosomes and plasma membrane at 3 lakh 20,000 G. The most appropriate use of detergents in the process of differential centrifugation of cells is to lyse the plasma membrane and extract cellular organelles. The organelle common to both prokaryotic and eukaryotic cells are mitochondria, chloroplast, Golgi bodies and ribosomes. It will be ribosomes. The bacterial cell wall is composed of peptidoglycan, cellulose, chitin and glycocalyx. So the bacterial cell wall is composed of peptidoglycan. The function of phenol red is a pH indicator dye. Okay, the function of phenol red cell media is a pH indicator dye. Any question, Harinarayana? If you have any specific question, then we'll discuss. If you have any specific questions from lecture notes or from your previous assignments or current assignments, you can tell. Synthetic medium and examples. Okay. So these are the different medias listed for bacterial growth. This is for yeast media, and this is for mammalian cell culture media. So, and the recipes. Okay, we have discussed. This is for insect cell culture media. synthetic medium and examples, what are the, these are the examples, Harinarayana, like DMEM, MEM alpha, these are the examples of this mammalian cell culture. And then this is the examples of yeast medium. Okay, you have to name this media's name, like name as an example. MEM will be a synthetic media, then RPMI, MEM alpha, RPMI, these are the difference. How can we recognize as per as definition? I'm not sure about your question, what you are asking. So basically, synthetic, what is defined or synthetic media? So basically, uh, synthetic media are very like highly purified and very defined chemical components compound will be there. Okay, like it will have peptone in nutrient broth, like we have we have in example now. So these are compositions are very much defined, so they are called synthetic media. Okay, and media which doesn't have any defined composition, those are called non-synthetic media. So you have a proper like you can synthesize it, right? Because you have those components written out there. So that means those are synthetic media. And non-synthetic media means those doesn't have any proper, like they have some imprecise compositions of ingredients, those are called non-synthetic media. Okay, I hope it's clear now. Defined means the composition you are sure of, you can synthesize those media, so that is why it's called defined or synthetic media. So those all are examples of synthetic or defined media, because you have those compositions written out. So today's class was short, because yesterday, like previous class, we already talked about week. You can unmute and speak, Harinarayana. Like your voice is not audible till now, but you can try if you want to speak up. But I'm not able to hear your voice. Maybe is there any problem from your side, because your voice is not audible? It's okay, you can, if your mic has some problem or some technical error is happening, you can just write down, because I can see that you are unmuted, but I'm not able to see your voice. So the cell culture part 2 I'll be discussing in this next class. If you have any specific doubt, you can just let me know, or else today's class was short because we already talked about those affinity purification last class. So week 7's content, basically the contents of lecture, was already covered in last class. So it was a lengthy class last week. This week we have talked about those assignments of week 6, and week 8 some examples of assignments, and we have taught about the cell culture, those parts. Further, if I have any doubt, please you can just uh, say also by unmuting yourself, or you can write down. So for week 8's assignments, you can now see that these are the possible this kind of questions you will be asked. So please go through those lecture notes carefully and download those lecture notes. Like, these are the lecture notes from Professor Vishal Trivedi. Download those and read. And also this as like lecture tutorial classes, if you have missed anything, visit the NPTEL lectures by Ananya YouTube channel and go through those videos. And also this lecture slides you will be able to get from the folder which is already there in the NPTEL portal. Madam, how are 9, 10, 11, 12 weeks? Are they similar in this way? Like, in which way, Harinarayana? 9, 10, 11 weeks will be similar, like how you were getting this lecture notes and all. It will be like that only. So you'll be having those lectures, and then I'll be like clearing your doubts in solving assignments, but not like assignments whose deadline is over. Those assignments I'll be solving, but if you have any doubt on live assignments, you can tell me. I will clear your concepts out. So all are same only, 9, 10, 11, 12 weeks. So I think you have already registered for the exam, and you were preparing for that. So for that, the lecture notes please go through, because everything will be from there. Clear your concepts. Anything you have doubt, please feel free to get your doubt cleared in this tutorial sessions. Okay. So next class we will be discussing week 8 cell culture medium part 2. So week 8 and week 9, and we'll be discussing some assignments from week 7, week 8, and so on, and some previous year. I am also discussing previous year's possible questions, so which will be useful for your exams and assignments. So this is all for today. Thanks for joining. See you on next week on Tuesday from 6 to 8 p.m. Thank you all.